Okay, this is part 2 for our beginning of chapter 5 lecture notes on energy flow in the cell. And in this video, I will be discussing, starting with point 4, on how enzymes make substances react. And I've got my handy dandy little chart over here to help us. It says enzymes speed up the rate of chemical reactions, and it's called being catalytic. We need to speed up the rate of chemical reactions. What they'll do is they'll recognize the molecules, they'll bind to the molecules, and then they'll alter specific reactions. They don't work for everything. It's a lock and key model, so one enzyme works for one reaction. Enzymes will remain unchanged in that reaction, and therefore they can be reused over and over again. And enzymes might be proteins, or they might be RNA molecules that other molecules will bond to, and then they'll work with the reaction. Our next point, enzymes will lower activation energy. And activation energy is the energy required to start a reaction. So here's my handy dandy little chart. You've got energy on your y-axis. You've got your progress of reaction on your x-axis. So reactants like glucose, let's take glucose for example, it will break down into its smaller parts because that's increasing in entropy. But normally for that to happen, you have to get it to a transition state. So if you want to burn glucose, literally you have to heat it up to make that happen. But in our cells, we can't heat up glucose enough to cause it to burn and change that way safely. We can't do that in a safe manner. So we have enzymes through a multi-step process that will break down the sugar. It will bond to an enzyme, it will break it one part, it will bond to other enzymes that will break down further, and so on. So what the enzymes do is they will bond to your reactants. This free energy is energy of a reaction with or without the enzyme. That means that this energy threshold has to be present no matter what. It will be there. It has to be reached. And then the enzyme will bond to it. But when the enzyme bonds, the energy needed to change it from this initial state to this transition state, this new mark, is going to be much less than without the enzyme. So over here on the right-hand side, it says activation energy of uncatalyzed reaction. That's going to be a lot greater than the activation energy required for a catalyzed reaction. So it'll have to increase that energy threshold a little bit, and then they hit the transition state, and then something will happen. In the case of glucose, the glucose will break apart. In the case of building the glucose, you have carbon coming together to form the glucose. And then you burn up or you release the energy, whichever is happening, and you end up with products at the end of the reaction. So let's go through our notes. Enzymes first. Number 1 will bind to the active site of a substrate. Number 2, enzymes help the reactants reach a transition state by, first of all, they help substrates get together, so they're not floating randomly in the cell. They are at the same place at the same time. They will orient the substrates in positions favoring a reaction. Again, think of these as three-dimensional molecules. You've got to get the right side of the Legos fitting together or they're not going to go together. So they get them in the right orientation for that reaction to occur. They also shut water out because remember, water through condensation reaction or hydrolysis reactions will change the molecules. So they only allow the water in that needs to be in, or allows no water in depending on what kind of reaction is going on. And it also induces changes in enzyme shape, so induced fit model. Sometimes when a substrate bonds to it, the enzyme will change shape to allow another substrate to bond to it. So enzymes will cause these things to happen in order for the reaction to take place. Enzymes can't work alone. They have to have something else with them. That's what that statement of enzymes don't work in a vacuum actually means. They need help from cofactors, which will bind to the enzymes to make them more reactive. And cofactors are non-proteins. This might be inorganic metal ions such as iron, or they might be your NAD+, your NADP+, or your FAD, and you'll see those when we do our Calvin cycles and other parts of cellular respiration or photosynthesis. Those are coenzymes that help those enzymes do their job. There's also controls over enzymes, so enzymes don't just keep working over and over because they're reusable, so they're always present. So how do you shut down a reaction if you don't want it to work anymore, or how do you speed up the reaction maybe if you want it to keep going. So there are different controls that help to cause these enzymes to work the way they should. Allosteric enzymes have regulatory sites on them which allow control substances to bind. So your body might produce a control substance that causes the enzyme to stop working, and it'll do that as it gets signals that it has enough of whatever product that enzyme helps to create. If it has enough of that product, it might make this it might make a control substance that will bind to this site on the enzyme, and it will cause the enzyme to change shape and stop working. So it kind of is like, whoa, we're good. Puts a hand over the reaction and gets it to quit. There's also feedback loops. So you have something called feedback inhibition, which after you've made so much product, the excess product or the extra product will then bind back to the enzyme, or an enzyme earlier in the pathway. Like if it's a 3-step pathway, maybe your final product in the 3rd step will go back and bind to an enzyme that caused the 1st step to happen. And so then it will shut down the synthesis of the product. So it'll cause the product to stop being created. So that way you don't waste time and waste energy and waste resources making a product that you already have enough of. Other things that can affect how enzymes work are temperature, pH, and salinity. So high temperatures can result in denaturation, which is a change in shape of an enzyme. So remember, enzymes are proteins, and as proteins get heated, they can change shape, or denature. So a high temperature, such as boiling something, might cause denaturation of the enzyme. Also, most enzymes operate at best at a pH near 7. Your body is really close to a 7. It goes 6.8 to 7.2. Anything greater or less than that can disrupt enzyme shape and function. And salinity must not be higher than the tolerance range, which salinity is the amount of dissolved salt in something. So it also has to be within the range of the enzyme, or again, extra sodium and chloride ions will bond to different places on the enzyme and cause it to change shape if there's too much of it. So that is your energy notes for chapter 5.